pink sickness, and the numbers of CC flies dwindling in as the Africa warming too much in Zambezi valleys, for example. This is the example of how the climate change affecting every single life on this earth. All right, ladies and gentlemen, may I introduce you our UMT, what we call it, a climate change adaptation lab, where I newly established this one for the past 2 years now in operation of this to validate actually what happening in the wild, and we can bring to the lab scales in order to validate according to their response, the adaptation of the tested organism through the IPCC various IPCC parameters, okay, in order to understand more on the physiological effect of various especially commercially important cultured animals in the aquatic media, that we can test them how they respond to the changes, extreme changes of the temperature or salinity or pH, in order for us to understand more on the adaptation behavior towards the climate change. This is important studies in order to, because it's very difficult to study on site in the wild, but it's only happen to validate it with the control parameters in the laboratory scale. All right, so this is part of our recent last 5-year studies, where we can see the temperature so much affecting the commercially important species, all right, to the environment in captivity. For example, what is their response, the maximum temperature response, and for, as you all know, the 1.5 degrees increment in temperatures can change so much in terms of physiological change of the aquatic animal as well. So this is very, very tough. It was before 4 degrees Celsius. Now the IPCC put it more stringent threshold level for 1.5 increment, 1.5 degrees Celsius increment that can cause significant, changes in the physiological or biology of the aquatic environments of the aquatic animals. Sorry. Then, of course, it further about the high temperature effect on the induced hybrid groupers. You can see how it responds to the temperatures in terms of the physiological parameter like liver proteins and muscle proteins for the species to react with the changes of temperatures from high to low or low to high. Then, of course, in terms of at the cellular level, we observe histologically on the effect of the high temperatures on the gill plasticity in our freshwater species, ikan jelawat, the kingfish, oven carp, and of course, how the increase in temperature affecting the fine structure of the gill, that affecting their physiological behavior and change, and of course, in the end, can retard in growth and even can cause the fish kills in captivities. Right. So it's further study on the effect of the acid pH, how it happen when the aquatic acidification happen, how it actually affecting the physiological condition of the fish, through this fine structure observation of the gill structures over the effect of the acid pH. Okay. And then, of course, the high temperature in the plasma glucose mobilization in the fish as well. We study on the gourami, how the plasma glucose react on the increase in temperature, where the higher temperature increase in plasma glucose, which is not good for the fish growth, right? And of course, it indicate the stress symptom with the extra energy requirement for their whole life. Okay, and of course, the high temperature also reduce spawning performance of the gourami. We found that where in terms of spawning capability, high temperature decrease the spawning capability significantly. That tested in captivity. Sorry. So of course, in the shellfish as well, for the mud crabs, all right, climate-induced water temperature change on the thermal tolerance, thermal polygons, and as expressed by the change in the growth, development, thermal resistance, maturation, spawning, and embryonic development throughout the recurrent crab lifespan. As we all know, all brachyuran or invertebrates, that the shellfish undergo the ecdysis for changes of their skin, and then from time to time, and this will affect when they are in the soft shell condition. They affect so much in terms of the thermals, thermal factors affecting their life during the changing ecdysis process, okay, over their lifespans. Okay, then of course in terms of the experiment that we have already studied on the thermal tolerance has been done very, very extensively to understand more on the physiological adaptation for this different lifespan, which is very complicated as compared to fish, because fish don't have changes of the skin, but for the invertebrate brachyuran crab, they have to undergo this changing of the skin, ecdysis process, and the metamorphosis process, and that is where the problem of the temperature and any changes of the water quality affecting their life and growth. Okay, for the thermal tolerance zone, for example, we found that for the shellfish, the best temperature is between around, you can see the thermal tolerance zone here, between 15 to 40 degrees, towards 40 degrees Celsius, thermal tolerance there. I can see the best tolerance around that, and we can draw the middle line here in order to have a optimal growth according to the temperature acclimatizations. All right, ladies and gentlemen, we come to the challenge during the COVID-19 pandemic. I guess this also experienced everywhere globally in the world, where the implementation of the movement control or the MCO to reduce the rate of COVID-19 infection occurs everywhere, including Indonesia, for example, Malaysia, Thailand now, and everywhere. Started in US, where the C-19 infect decision were based on the Malaysian national security here, very strict, control, and of course, coupled with the Ministry of Health Malaysia and database to curb the impact of C-19 virus and strategy to support essential industry. So of course, this is the current situation now, where actually Malaysia, Indonesia, we have around on average of 15,000 cases per day, and it's fluctuated from time to time, depending on our government is trying to get whole Malaysian to be vaccinated the soonest as possible in order to protect them from this pandemic effect, right? So the impact of this COVID-19 on the aquaculture industry has been so tremendous. For example, so of course the thing that when we cannot work, where the people cannot go out from their home and work in the field, of course will affect substantially on the on the supply chains, on the raw material supplies, and of course in the hatchery, in the grow out pond, of course it affecting the growth of the fish, and sometimes can cause the kill because of not so many people can handle the case due to the pandemic, and of course to their market chain, transportations, and market supplies, exports to the neighboring countries or to other countries, also largely affecting this change, for example, in terms of the supply chains. Okay, this is very, very important for us to tackle from here, how to solve the problem. And of course, the Malaysian government actually give more lenient approach to enable the workers in this aquaculture to work, but still limiting limited numbers of the entry to these industries due to the to these pandemics concerns. All right, so the impact of this C-19 on Malaysian aquaculture sector revolve around two crucial components. For example, in the significant reduction in domestic and global demand, breaking in the seafood supply chain. This is more important because from the survey we found that the general impact, everybody agree about 90% agree the general impact of the C-19 in the aquaculture industry. And of course, in terms of the production, decrease in sale, increase in sale, for example, very small numbers, the stockpile out due to low demand, decrease in price of the cultured animal due to the oversupply because of the no, nobody taken it due to this scare of pandemic to get out. And of course, the demand increase in demand, but decrease in demand as well. Okay, see, 70% agree with the increase in demand and 20% decrease in demand, for example, for this. And of course, the logistic bottleneck where 70%, affecting the synergy, affecting the logistic bottleneck, for example. Then, of course, the bureaucracy-related problem almost draw. Then, employee-related problem, people cannot work. Okay, about 69%, and of course, remaining there. That's why limiting entry of this in the Aquaculture industry allowed by the government. Okay, temporary closure of the fish mill. People agreed, 52% agreed, and 45% say no. Okay, that is. All right. So COVID-19 on Malaysian aquaculture, actually based on that, multiple type of the assistance needed by the respondent, for example, is sales services, 56%, finance assistance, of course, the most, about 70%, require financial assistance and technical service assistance, about 36%, of which this also to transfer the needs for the help, the need help for this for the industry from the government. In fact, type of the multiple assistance needed to study the study of the impact C19 on the aquaculture industry in Malaysia. So this is the thing, right? Okay, conclusion remark for this. While searching on the short-term CCA effect, this model is applied for long-term mitigation measures towards sustainable aquaculture production and moving towards IR 4.0, which is very important for the IoT facilities in the new era in aqua farming to secure productivity with limited labor, for example. And of course, the IR 4.0 has been developed everywhere in this world, and the IoT in order to facilitate, the aquaculture industry has been well developed from time to time. So under the pandemic situation, the financial package, government provide the financial stimulation package for certain sectors, and of course, Malaysian government also facing the very limited problem of the finance, for example, in order to provide all. We are not a rich country, for example, in order to provide all the package for the pupils in Malaysia, and of course has to be a targeted, industry in order to secure the food, security for the nation, for the next generation. Of course, in terms of food security is very important. Where in aquaculture industry plays very, very important roles in order to ensure continuous supply of food in the future. Okay, I think that's all from me. Thank you so much. It's my great pleasure to meet you today. Okay, thank you.
Okay, thank you, Professor Mazlan.
Yeah.
For the presentations. Interesting topic on climate change and aquaculture. Me myself also work on climate change along with Professor Fredolin Tanggang.
Oh yeah, my colleague in UKM.
Yeah,
I was from UKM before. You asked Muhammad, he knows me very well. I'm his boy.
Yeah,
Dr. Muhammad.
Dr. Muhammad was your former student.
Yeah.
Yeah. Hope we could have a good collaborations in the future.
You are most welcome, Dr. Yopi, or Unsia can, maybe the rector can communicate with me.
Okay, thank you very much.
Prof. Samsul Rizal, so that we can have the signing of the document to work further. I'm most welcome.
Thank you very much, Professor Mazlan Abdul Ghaffar of the UNT. Hope we could have good cooperations in the future between UMT and Syed Kuala University. Okay, please give applause to Professor Mazlan Abdul Ghaffar. Thank you very much for your time. Okay.
Thank you.
Should the participant have any question, please write it down in the chat room. I would like to remind you that we keep the questions to be answered until the last speaker. Okay, now we proceed to the next speaker, Professor Junichi Tanaka of the Ryukyus University. Good afternoon in Okinawa, Professor Tanaka.
Hello.
Hello.
Do you hear me?
Yeah, I can hear you clearly. Okay. Good afternoon in Okinawa.
Yeah, it's noon already.
Oh, no, good day, I think.
Yeah.
Yeah. Okay, Professor Tanaka is a professor in the Department of Chemistry, Biology, and Marine Science, the Ryukyu University, Okinawa, Japan. His expertise in the field of natural products. Correct me if I'm wrong. Okay.
Yes.
Professor Tanaka, the presentation time will last for about a half hour, 30 minutes.
Yep.
Okay, you have plenty of time to share your knowledge.
Okay.
Okay. In the next 30 minutes, the floor is yours. Please, Professor Tanaka.
Yep.
Okay. Host, please help Professor Tanaka's PPT to be opened. Okay, Professor Tanaka.
Do you can you see my slide?
Okay, we could see it clearly. Please carry on.
All right. Let me start.
Okay, thank you.
Okay. Anyhow, thank you for having me on this symposium. It's a great honor to be a keynote speaker. I appreciate the organizer to have this symposium even under the spread of COVID-19 in Indonesia, Japan, and everywhere in the world. On this opportunity, I like to talk about introduction to marine natural products, because I suppose the majority of this symposium audience may be faculty member or graduate students of marine science or fisheries. So they are not specialists in marine natural product chemistry. So I hope you understand my contents as one of the field of marine science or chemistry. Sorry, I don't know why it doesn't go further. I'm very sorry. Why?
Are there any problems that we could help, Professor
Yeah,
Tanaka?
seem to be problem.
Okay. Yeah.
I don't know why.
I think House could help Professor Tanaka to cope with this problem. The presentations does not work, I think.
It works, but now I don't know.
Okay.
It does not go.
Okay, it works now. We can see it here. Okay,
all
we
right.
can see your presentation.
Sorry for the interruption.
Please carry on, Professor.
In this presentation, I'd like to talk about first on the introduction to natural product chemistry, brought by organisms and molecules from some species from Indonesia, chemical and biological aspects of the molecules, and additional matters on the marine natural product chemistry. In the field of marine natural products, we usually focus on so-called secondary metabolites. They are produced by some organisms, but not all. Usually, they may be classified as polyketides, alkaloids, terpenoids, macrolides, dependent on the molecule skeleton. But they are not. In addition to them, there are some other molecules like the polymers, like polysaccharides, such as agar or carrageenan, the latter which is produced in Indonesia. And also there are peptides and proteins like venomous toxin, like the one from produced by Consner, which is one of them became analgesic drug already. Okay. Natural products are metabolites from organisms, and we utilize them from ancient time until now. Probably most of the audience is familiar with jamu, and jamu utilize natural products from plants. though not pure state, but as a mixture. Marine natural product chemistry is a part of natural products chemistry, and especially focused on marine resources. Marine organisms are relatively new sources when you compare from terrestrial plants or microbiome. But it started around 1960s or 70s. And the later Professor Paul Sawyer is one of the pioneers, and also he was dubbed as the father of marine natural products. Anyhow, in the last 5 to 6 decades, a large number of unique bioactive and also precious molecules have been found from marine organisms so far, and new molecules have been discovered every year. When you talk about a precious material from the sea, you may remember that of sunken treasure, like gold bars or ancient artifacts. But if you compare the price of gold and one of the most expensive molecules shown here, found from a sponge near Tokyo, you may realize that this kind of compound is quite expensive. But the demand is totally different. Maybe everybody wants gold, but only some special biologists need this kind of molecule. Anyhow, there might be more molecules like this in Indonesian waters. But to discover this kind of molecule, in addition to luck, you need intellectual skills to search and identify such a molecule. So science education, training on chemical and biological research, and maybe additional resources are important. Okay, now next is a starting point of natural product chemistry. I'm based in Okinawa, located southwest part of Japan and close to Taiwan. The distance from the place where I live to the westernmost island in Okinawa is only 500 kilometers. But when you compare the size of Okinawa with Indonesia, apparently Indonesia has much wider area and many more habitats for marine organisms. So you can expect a lot of diverse organisms because as the eastern part of Indonesia is a center of biodiversity, though Indonesia is located western end. During my experience, I feel that marine areas with many islands provide various habitats. And by changing a little distance, you can find difference of fauna. As Indonesia has about 17,000 islands, right? Probably you can also expect a lot of diverse organisms by changing the visiting place. During the late 1990s and the first decade of 21st century, I had the opportunity to visit Indonesia under several collaborative projects in Indonesia and Japan. My collaborators are LIPI, Oceanology, UNIRA, and UNDIP, and Osaka University in Japan, and Panama in Spain. And I have been to the location with these 5 flights for exploration. And I was happy to observe various unique organisms which I cannot see in Okinawa. These are palapa, green alga, soft coral, probably bryozoan, and polychaete. You can also find unique chemicates like this in forests and reactive areas. Before going further, what you need to know is the basic knowledge from this field. Dr. Hanif Nobriande Hanif is a lecturer at the chemistry department of IPD. He wrote a review on marine natural products from Indonesian waters. You can freely download this material. You can find what kind of molecules discovered so far from Indonesian marine organisms, and you can be familiar with them. But there are several questions on this aspect. Not like terrestrial plants, the same organism may be found at different countries, as in the following example. The same molecule can be found from different organisms. And in some cases, in some marine invertebrates, there is a possibility that the same organism is reported under different scientific name. Once there was a joke that if you separate one specimen into two and send each to different taxonomists, you may receive different names. It makes confusion. Anyway, and in addition, not to waste your time and resources, the replication, it means not to repeat identification process of non-metabolites is quite important. In addition, when you encounter new molecules, you also have to use some commercial databases such as Marine or SciFinder, though we have to pay. Okay, this is an advertisement of Nobriandi. Dr. Nobriandi organizes a summer school of marine natural products from 2019. Unfortunately, it was cancelled last year due to COVID-19, but he organized another one this year, this month. Although the registration was closed in July, but he extended it until August 5. So if you have any interest, you can check. All right, back to the exploration in the field. You have to recognize some common or cosmopolitan species which can be found throughout Indonesia and surrounding area. Here are some examples with a photo and also molecule. This is so-called just spongia testinaria, and any diver can recognize it if there is, because it grows 2 meters or so. More examples. This sponge colors the vivid yellow inside, while this sponge can be used for bathing sponge. Additional examples. This unique shaped sponge is known as ion cell basket, and a number of brominated phenolics are reported. Just sponging a unique alkali with this kind of structure, and one of them is sold as a precious reagent. On the other hand, there are endemic species which can be seen only at certain areas. The Candida spongioid up in the photo found at one area contains a series of quite toxic macrolides named Candida spongioid, but which was originally reported from the same sponge collected at Papua New Guinea. I did not see it in except in Indonesia, but apparently the distribution is not limited to Indonesia. Betungoids are named after the collection site, Bitung, in North Sulawesi. The sponge looks like Selner swimfly, very well-known sponge in this field, but the morphology are a little bit different. More endemic sponges and their metabolites like this. Okay, and these endemic sponges can be collected only small amount. Can be collected only small amount, but nowadays, thanks to the instruments, you can illustrate the structure with modern instruments. But endemic organisms do not always contain new metabolites. These organisms are rare and cannot be found everywhere, but they were not attractive in a chemical or biological studies. Next, I'd like to talk about what to do, what to use for doing the research. Vicky Kurnianda is a faculty member of University of Shakora and also currently my graduate student for PhD. Soon he will go back to Indonesia, I guess. Only a week ago, he told me his dream that he would like to have a sophisticated instrument. Just by injecting a small amount of extract, and you get the information on the chemistry and also biology. But judging from the nature of chemicals and also biological activity, it is not likely to be developed soon. To characterize molecules of your interest, you have to use several instruments. Usually, we cannot obtain pure chemicals from organisms, so we have to separate or purify the molecules. For that purpose, there are several kinds of chromatography. Once you obtain pure chemicals, it can be analyzed with mass spectrometry to check new unknown molecules with the formula weight or molecular formula. X-ray crystallography is good for crystalline molecules to confirm the planar structure and also to determine its chemistry. An example is the determination of a downstream metabolite named X. This natural compound was illustrated by NMR force, but due to signal overlaps in NMR, we face difficulty on the determination of its chemistry. This natural compound was illustrated by NMR. And after my student tried to make several derivatives, and she found this dihydrox group molecule derivative crystalline, and the chemistry was so as shown here. NMR is the most useful instrument to analyze unknown molecules. An example is another molecule named franklin Y, a stereoisomer of franklin X. Also, the signal of the complementary compound were observed as in this figure. We were not confident on the whole stereochemistry due to signals were observed closely. So once again, my student made several derivatives. One of them was found to show separate signals, and we determined the whole stereochemistry as shown here. Nowadays, in addition to the instruments, calculation study is also an indispensable method to assist structure elucidation. This is a case of two new sextatenoids from an Okinawan sponge collected at tip of coral reef. This linear molecule must have a flexibility on the structure, and there is only one hierarchy here. After obtaining many conformers, each enantiomer was calculated and compared. Then we get conclusion. For another molecule from this sponge, there were two candidates structure. But when we study, it's a kind of NMR measurement, and also calculation study. We reached to the conclusion that the left structure is the answer. For biological evaluation, many labs use clean bench or safety cabinet for handling cultured cells or microbes. Actually, equipment and microplate reader can be used not only for biological assays, but for some assays with chemicals. For more sophisticated research, you may attend a lecture of Professor Arai of Usk University this afternoon at room 1. But some student or young faculty member in University of Sekolah or as a part of Indonesian university may feel that you cannot find some of the things in your lab. Don't worry. We had a similar experience back in 1980s and 90s. We used to work without NMR, mass spectrometer, or crystallography, etc. We could not make many papers, but several. Why? Simply by asking your collaborators to run data and evaluate biological activity. For example, I heard that there are only 3 NMR machines with superconducting magnet in Indonesia, and there is a long queue to obtain the data. But there are a large number of NMR machines in the world, and some of the machines may have idle time. But everyone has limited time to do research. What you need to do is to find good problems. Okay. In the final part of my talk, I'd like to discuss another aspect briefly. During the research on my natural products, you may encounter various phenomena. A unique alkaloid named manzanine 8 was originally reported by mentor, late Professor Higa, and my former colleague Sakai from Okinawan sponge named Harikroda. They also found manzanine A and analogs from a different sponge, Arkansas strongylophora ingens, which spreads widely. The name of the compound was taken from our favorite collection slide named Manza in Okinawa. One of the sponge, Arkansas strongylophora ingens, can be found in Indonesia widely from Sabang on the west to Sorong in the east or Icfaes in the south. The morphology also differs at some sites, and you can expect that the chemical content may change as the morphology. Actually, researchers at Unsura and Kumamoto University in Japan reported new analogs isolated from a specimen of Arkansas strongylophora collected at Manila. In addition, information technology changes the research style of natural product chemistry. GNPS, or Global Natural Product Social Monitoring Networks, developed by Professor Bresstein of the University of California, San Diego, has been utilized by many research groups to analyze the diversity of metabolites or to find new chemical entities similar to previously known. Anyone can use this database software, but you have to prepare MS data of the pure compound and mixture like Icfaes. An example shown here is our data analyzed by Vicky on the chemical contents of Pyridia milligram. As you can see, the extract can be separated roughly into 2 groups and a small subgroup. And the topic is not new at all, but I like to show it as a means to explore further new organs from Indonesian waters. Mesozoic coral ecosystem, or MCEs in short, can be taken as deeper coral reefs. According to some researchers, it may be below 30-meter depth, but shallower than 150 meters. Due to the global warming, shallow coral reefs experience a coral reef bleaching event. But deeper coral reefs can be taken as a kind of refuge of stony coral species. The number of species in deeper area is probably less than that of the shallow coral reefs, but there still exists a huge number of organisms. Due to the limit of scuba diving, most research groups did not collect specimens as those of the shallow waters. Scuba was introduced after the World War II, and it dramatically changed the research style at shallow waters. However, there is a limit with scuba if we want to go deeper. Nitrogen narcosis affects you below 30 meters, and you may not be able to think properly. Oxygen toxicity may happen below 70 meters or so, and it may kill you. You cannot stay long at deeper water with scuba, but you have to stay long at shallow water before exiting, not to get decompression sickness. So we use thermic revisor diving instead, simply replacing excess nitrogen and oxygen with helium. And you can search newer marine organisms, mainly in deep upper mesophotic zone, say 50 to 60 meter range. We started to use it from 2005, and so I show you several results. There are a few more, for example, molecules isolated from mesophotic coral ecosystem. Some sponges like this, Icfaes japonicus, can be found only below 45 meter depth. Many sponges were taken at the same site, but until my exploration, it remained to be poor. So maybe the same thing happened to other places. Another topic is nowadays, researchers have interest on the biosynthesis genes for secondary metabolites to understand how these complex molecules are made. In 2004, two pioneer works appeared. One is On My Day by John Pierre, and another one is Biosystem 1 by Mark Taylor. And then, thanks to the development of the next generation sequencers and other information technology, more molecules, biosynthetic genes of more molecules are revealed. But still, there are many more interesting molecules to be investigated. And immersion as an alternative method to natural harvest of marine organisms, people aim to produce molecules by using the identified biosynthetic genes, but it is a still big challenge. All right. I'd like to summarize my discussion as a suggestion. Indonesia has been known as a center of biodiversity, and you still have a lot of resources and marine organisms. Starting from knowing molecules and species, you need expeditions, instrumentation, having good researchers both in chemistry and biology. In addition, you have to keep collected specimens in a good manner, preferably deposit at a place like Natural History Museum, where curators work. And we have to make publications to respond to the demand of funding agency and public. All right, that's all for my talk, and thank you for your attention and patience.
Thank you very much, Professor Tanaka, for lecturing us, for giving us such informative and interesting presentation. At Syekh Kuala University, we have Professor Musri and Dr. Vicky Kurnianda, who are also concerned on marine sponge and marine natural products. Hope we could have a good cooperations between Icfaes University and Syekh Kuala in the future, publishing papers together, I think.
Hopefully.
Yeah. Professor Tanaka, thank you very much for your time. Give big hand to Professor Tanaka. Okay.
Thank you.
All right. We invite the last speaker. Please welcome Professor Fujaya of Hasanuddin University, Makassar, Indonesia. Professor Fujaya, Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning in Makassar, I think. Still morning?
Yes.
Oh, good day. Good day, Makassar.
Good day. Yes.
Okay, Professor Fujaya, you have for about 30 minutes 30 minutes or a half hour for presentations time. Okay. All right, Professor, the floor is yours for about 30 minutes, please, Professor Pujaya.
Thank you,
Yeah.
moderator. Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The Honorable Mr. Chancellor of Universitas Yakuala, Mr. Dean, Mr. Chairman, and all of the committee, thank you for inviting me to share my experience and knowledge in this valuable conference. Thank you to the moderator for introduction of me. I greet Professor Masan and Professor Tanaka and all of the participants. Let me start my presentation. I will to share screen. Okay, is it visible on your screen?
Yeah, we could clearly see your presentation.
Okay.
Okay, the voice is also clear. Please go ahead, Professor Pujaya.
Thank you.
Okay.
Ladies and gentlemen, today I'll talk about triobiotic for sustainable aquaculture. For this talk, I have 4 steps. Firstly, I will give a brief introduction to sustainable aquaculture. And secondly, we'll explain what the triobiotic is. Thirdly, I will discuss the application of triobiotic on Nile tilapia, Panama shrimp, and mud crabs. And lastly, I will give a conclusion. Ladies and gentlemen, sustainable aquaculture is a very challenging issue over the last few years, but what is meant by sustainable aquaculture? I think, and maybe you all think, that sustainable aquaculture is a situation where aquaculturists want to continue their cultivation activities for various reasons. Aquaculture sustainable can only be achieved when fish cultivators are happy because there is a lot of harvest, when fish cultivators can support their families properly from the results of cultivation, when the community does not lack fish to meet their nutritional needs, and when cultivation activities do not damage the surrounding environment that makes us suffer. Those are the real reason to keep doing aquaculture business, right? But unfortunately, cultivation activities that damage the environment are still being carried out, and as a result, the disease spread out of control. Diseases that attack the wild shrimp industry are the result of excessive use of feed and chemical drugs. And to stimulate growth, feeding a lot of high protein content is considered to be helpful, even though excessive feed will damage water quality. Poor water quality will cause and trigger a disease. And when the disease strikes, treatment for disease is mostly using antibiotics. Unfortunately, inappropriate use of antibiotics has eliminated good bacteria and created resistance to pathogenic bacteria. The disease is mostly associated with poor water quality, prodding, and improper handling. As well as in the shrimp farming, fish farming is also experiencing the same thing due to poor handling and application of aquaculture technology. Streptococcal disease killing a lot of fish in pond. Yeah, ladies and gentlemen and respected guests, there are 2 major complementary strategies that deal with bacterial disease. First one is reactive strategy, mainly focused on treatment, and the second one is proactive strategies focus on prevention. Reactive strategies usually using antibiotics in feed, but the main issues associated with these practices are antibiotic resistance and the presence of antibiotic residues in marketed seafood products. This case triggered a ban on the use of antibiotics and hazardous chemical in aquaculture activities. In this picture, we can see that antibiotic resistance is triggered by inappropriate using the antibiotic, incorrect dosage and treatment duration, and others. And then the antibiotics resistance worsens consumer health and harms to the economy. I think prevention is better than cure. And it's also cheaper. Disease can be prevented with good environmental management. Also, use natural ingredients to boost immunity and promote growth. Yeah, certainly we can keep cursing this incident, but it's better to find a solution. Let's take a look at the 5 main principles for sustainable aquaculture put forward by Professor Rohmin Dahuri, Chairman of Indonesian Aquaculture Society. But in this presentation, I highlight the 5th principle, namely the technology used in cultivation must be environmentally friendly. That is very good. Great hopes are aimed at these 3 biotics. That is probiotic, prebiotics, and phytobiotics. Products that combine probiotics and prebiotics are called symbiotics, where the combination of the 3 we call triobiotics. Let's get to know what probiotics are. Probiotic was first introduced by Lily DM and Stilwell H. at 1965. They describe probiotic as a substance secreted by one microorganism that stimulate the growth of another. Furthermore, it is said that the benefits of probiotics are related to nutrient contribution, to promote survival, to improve the host immune system, to promote growth and or antibacterial properties against pathogenic bacteria. And for aquatic environments, the function of probiotic is decreasing algae growth, decreasing organic load, increasing nutrient concentration, increasing beneficial bacterial population, inhibiting of potential pathogens, and etc. Popular probiotics microorganisms include some species of Bacillus, Nitrosomonas, Nitrobacter, Rhodopseumonas, some species of Lactobacillus and Saccharomyces cerevisiae, and many others. So, what is a prebiotic? Prebiotics are defined as digestible nutritional ingredients that have a beneficial effect on the host, promote the growth of beneficial bacteria in the gastrointestinal tract. Simply put, prebiotics are substrates or food for probiotic microbes. Prebiotic fermentation by good microbiota will produce short-chain fatty acid, including lactic acid, butyric acid, and propionic acid, act as an antimicrobial. Actually, prebiotics are natural present in food, such as asparagus, garlic, chicory, onions, wheat, honey, bananas, tomatoes, soybean, milk, nuts, seaweed, macroalgae, and others. Well, on the vegetable and fruit we usually eat. Then, what about phytobiotics? Phytobiotics is all type of plant contain active ingredients or substances that are useful for medicine. The use of phytobiotic for the health management of aquaculture organisms is a promising new alternative. Probiotics are not phytobiotics are not only efficient on the non-specific immune system, but also on growth performance. Yeah, Jenna et al. In 2018 reported that the various properties of phytobiotics, such as anti-stress, growth promotion, appetite stimulation, immune system enhancement, rootstock maturation, aphrodisiac, and antipathogenic due to the presence of various bioactive substances, alkaloids, terpenoids, tannins, saponins, glycosides, flavonoids, phenolics, steroids, and essential oils. In this slide, you can see that some probiotics as well as phytobiotics that are abundantly available in Indonesia, such as temulawak, or the scientific term is Curcuma santorisa, jahe or Cingiber officinale, and others. Yeah. And also in this slide, you can see there are just a few examples of phytobiotic. There are still many phytobiotics to choose for aquaculture and are easily available around us. Ladies and gentlemen, I'm trying to develop triobiotics from local ingredients, and I found that in addition to containing the active substance stated by previous researchers, this product also contains curcumin, some vitamins, minerals such as potassium, sodium, zinc, and others. Also contain digestive enzymes like protease, lipase, and amylase. This is the important study to test the triobiotic in Nile tilapia and shrimp and mud crab cultivation. You can see in this slide, tilapia responded well to the given triobiotics. There is a significant difference in response to the dose of triobiotics given, including weight gain, specific growth rate, feed efficiency, and survival rate. And you can see the best dose is about 100 milligrams to 300 milligrams triobiotics per kilogram of feed. And in trials in cultivation ponds that actually belong to fish farmers, the triobiotics are applied to feed and cultivation water, and the results are very promising. After 15 weeks of rearing, or about 110 days, farmers can harvest fish with an average size of 330 grams or 3 sizes per kilogram. More promising because the FCR obtained is only 0.5 to 0.9. It's very efficient. That because the provision of triobiotics in cultivated water has succeeded in growing natural food very well. In addition, the efficiency of using feed for growth also increased. Efficiency in the cultivation of feed can be seen from the intestinal histology picture. Oswald et al. Found that the supplement of phytobiotic extract in tilapia feed caused the intestinal villi to become longer and wider, which indicated that the absorption of nutrients was increasing. We also get the same result. And administration of curcumin was also reported by Ashley et al. They found that a decreasing in the total number of bacteria, exit fermentative bacteria, Vibrio spacious and fecal coliform. The bacteria usually make fish ill and don't grow optimally. And however, the addition of curcumin in the diet increased blood total protein and phagocytic activity. This indicates an increased immune response. This information is very, very useful. Our teams also tested the triobiotics on Faname shrimp and found that growth and survival rates were better than controls treatment or feed without triobiotics. Protein retention and energy retention were also improved, especially at the dose of 3 grams per kilogram of feed. Only fat retention was lower, but it may be good for low-fat shrimp development in the future. Field trials of shrimp culture with intensive systems also obtained promising results. At the age of 2 months, the shrimp has reached an average size of 17 grams or 59 ounce size. That is a very good growth. And if you pay closer attention in this table, you can see that the salinity continued to decrease until 2 ppt. It's because to heavy rains about 1 month rearing of shrimp. I think the shrimp can strong to maintain the stress due to the low salinity condition, probably due to the trace mineral content in this triobiotic, especially potassium and sodium that adding in their diet. As we know, potassium and sodium are important ions in the osmoregulation process. You can see in this slide, under condition of low salinity or high salinity, organisms become very active in pumping body fluids due to differences in osmotic pressure. In the seawater, it's hypoosmotics. However, in the freshwater, the shrimp is hyperosmotics. This is why potassium and sodium are needed to make active transport from water to the body and reciprocally to maintain homeostasis. In seawater, shrimp are hypoosmotic, so to maintain homeostasis, shrimp must hold water from leaving the body and pump out ions that enter the body through diffusion. The opposite happens when the shrimp are in fresh water. The active compound of phytobiotics also work through detoxification. The example in this diagram shows that phytochemical antioxidants act on gene targets and convert the ability of cells to maintain homeostasis during stress by inducing detoxifying enzymes to remove harmful substances. Ladies and gentlemen, this triobiotics has also been tested on crabs. Although the disease crisis in crab culture are not as severe as in shrimp and tilapia, this triobiotic is still needed to induce molting and growth. Crabs is one of the most popular seafood products, the demand for which is not accompanied by sufficient supply. The popular product of crab cultivation include jumbo crab, fat crab, and soft shell crab. And to produce this product, the most common cultivation activities are growing out in the pond, fattening, and soft shell crab production, and usually rearing in confinements. Because of that, crabs often don't want to eat, don't grow, and don't molt, maybe because of stress. Therefore, we need to find a new way to get them to eat, molt, and grow in a cultivation environment. Yes, molting is a precursor growth in crustacea, including crab and shrimp. To get big, crabs have to molt first. So far, one of the most widely used to simulate molting is lime autotomy. I don't think this method cares about animal welfare. In fact, this method often results in the appearance of the product that is undesirable, such as losing the legs or the legs growing small and abnormal and causing the price to fall. I think and I believe that many of the conference participants here think that the lime autotomy method is not suitable for the production of soft-shell crabs economically and ethically. So we need to find methods that are more friendly to animals. Okay, first let's see why crabs need molting and what regulates it. Because crabs have a hard exoskeleton that surrounds the outside of their bodies. This hard shell cannot expand as the crab grows. So periodically, the crab must shed its shell and develop a new and bigger shell. The process called molting. The molting process is controlled by the molting hormone, namely ecdysteroids. This graph explains well the dynamics of the molting hormone during the molting period. That is, ecdysteroid titers rise sharply during the pre-molt phase and decrease drastically just prior to ecdysis. Ladies and gentlemen, gladly, there is a chemical compound similar to the molting hormone found in plants. It's called a phytoecdysteroid. According to Klein, this phytochemical also play a role in increasing protein synthesis. So we use this information to develop a method of stimulating growth and molting using theobiotics in crab culture. The dose of theobiotics affected on growth and feed efficiency of crabs. In this slide, you can see that the dose of 600 milligrams of herbal extracts per kilogram of feed gave the highest growth and feed efficiency. This slide also saw a striking difference between the growth and feed efficiency of molting and non-molting crabs during the study. Here it can be seen that in crab cultivation, molting management is very, very important. The frequency of giving theobiotics also affected the growth and molting of crabs. In this picture, it appeared that giving theobiotics 2 to 3 days a week gave a better result. Compared in the control treatment, it appeared that there were no molting crabs during the 30 days of the study. All right, ladies and gentlemen, lastly, I would like to conclude that first, sustainable aquaculture is necessary. Second, treating the environment and farm animals well will provide long-term benefits. Third, prevention is better than cure. And fourth, natural ingredients such as probiotics, prebiotics, and phytobiotics, whether applied single or in combination, are very promising to overcome the problem of cultivation. Finally, I want to say it's time to return to use organic material in aquaculture activities for sustainability. Okay, that's all my presentation. Thank you so much for your attention and listening. My talk, hopefully it may be useful. Thank you.
Thank you very much, Professor Fujaya, for sharing a good information to us.
Tidak usah direkrut. Jadi dari rekrut pun kita harus ketat. Lalu bagaimana?
Host, please keep it silent. Okay, thank you very much, Professor Fujaya, for sharing good information to us. the presentations talks about how to keep aquaculture activity sustained, I think. Okay, thank you very much for your time, Professor Fujaya. Thank you very much for your
Yes,
time,
most
Professor
welcome.
Fujaya. Okay. Okay, give big hand to Professor Fujaya. Before we continue to the discussions time, we would like to show video teaser of Icfaes for about 3 or 5 minutes. In the meantime, please prepare questions to be asked to the professors. Please go ahead, host. Okay, thank you very much. Ladies and gentlemen, we are entering the discussions time. Now come to the questions and answer sessions. Please raise your hand if you want to discuss directly to the professors. For the first session, we would like to have 3 questions who will communicate directly to the professors. by using raise hand facility in a Zoom meeting. Anyone who would like to ask directly to Professor Dr. Muhammadar, I think? Karina. Ibu Sofiatuddin Karina, would like to ask something? Would like to discuss something to Professor?
Yes.
Yeah.
Yes,
okay,
please.
Go ahead, Bu Karina.
Okay, thank you very much, Mr. Yopi. I'd like to give a question to Professor Tanaka from Japan. Glad to see you, Professor Tanaka.
Hi.
Yeah, it's a very interesting presentation from you.
Thank you.
Glad to hear you again. Some researchers nowadays have the complicated problems to separate the racemic mixture of the compounds from secondary metabolite. According to you, what is the direct or the complete method that we can use to separate this racemic enantiomer from the compounds? Thank you, Mr. Junichi Tanaka.
Please.
You are
Mr. Yopi.
Okay, please go ahead, Mr. Professor
You
Tanaka. Okay.
you are asking the question about to separate enantiomers. Is my understanding all right?
My question is about the method, how we can separate the racemic mixture of the compounds. Because sometimes when we found the compound of the secondary metabolite, we found the racemic mixture
Okay.
S and R, so it's complicated for us to separate it. Until now, I do not understand that. Please, Mr. Tanaka, if you have some method to yeah,
Okay.
to share with us. Thank you.
There must be several methods. One, probably the most common one is chiral, by using separation by using chiral, so-called chiral column, usually HPLC, and also there must be the one with GC also, but it depends on the nature of the molecule. May I ask what kind of molecule it is?
Ibu Karina, professor is asking what kind of molecule?
No, I just asking the molecules in general. Sometimes
for chiral columns, probably there is no column can be used for every kind of organs, molecules. So it depends on the compounds you are going to separate. If you need to separate direct as it is, you have to use this kind of chiral chromatography on HPLC. Okay. Some chemical companies sell this kind of color, and there is some specific solvent used for this system. And if it is just a matter of knowing the ratio of enantiomers, that is another matter. If you, in that case, for example, like determination of amino acid composition, you may use, you may attach some other chiral molecule to it and just to separate normal HPLC column, and you can find out the ratio.
Thank you very much, Professor Tanaka, for
Thank you, Professor.
for the explanation.
Welcome.
Thank you very much, Ibu Karina, for joining us in asking the questions. Okay, are there anyone else who like to ask or communicate directly to the professor or professors here? Dr. Muhammadan, I think, would like to ask something or discuss something to Professor Mazlan. Dr. Muhammadan, you may use a raise hand facility. Yeah.
Dr. Muhammadar via chat.
Okay, perhaps our chairman, Dr. Dedi, perhaps you have questions to ask to Professor Mazlan or one of our professor here.
IOP, there is some question in the chat room. Maybe
Oh yeah, many questions in the chat room, I think.
Dr. UP can read the question, yeah.
Okay, yeah, we could read the questions. I think questions to Professor Mazlan. I think, what is the effect of climate change on germplasms, and what are real examples that have occurred that can affect the fisheries economy? Professor Mazlan? Professor Mazlan?
Oh yeah, yeah. Thank you very much.
Okay.
Yeah,
You
on the
may answer
germplasm,
the questions. Okay.
on the germplasm, I'm not very clear there.
Okay, from Dr. Muhammadar, he is asking what is the effect of climate change on germplasm, and what are real examples that have occurred that can affect the fisheries economy?
Oh, okay, okay. It's a very good question, you know,
Okay.
from Dr.
Questions in the
Muhammad.
chat room. I picked one
Yeah,
of them.
yeah,
Okay.
yeah, I know. Right. As I mentioned in my slide presentation, the fisheries economic affected by the climate change, and of course, is actually can be considered as the intangible impact, that we because it is the secondary effect after the climate change parameters, the extreme weathers that affecting the productivity, what causing the decrease in productivity due to the effect, on the of this unfavorable impact of these parameters onto the physiology. For example, that's why we study very, very micro level to understand what changes in terms of their gill fine structure. For example, my feet in the gill fine structure studies, and of course on the, for example, on the oxygenation rates for their body, the same as, for example, when you observe on the COVID patient, they are monitoring on the oxygen using the oximeters, the level of oxygen in our body. So when there is a very, very unfavorable environment happen in the captivity, because in the wild, it's very difficult to measure, because the fish or the animal can avoid the unfavorable condition. So the key words here is actually favorable condition, where when there is that happen to the disturbance of the climate change parameter, for example, in acidifications, in lowering the pH in the environment, where the fish in captivity that we culture, for example, unable to escape from the unfavorable condition. So this will affect their biological, in terms of genetic, in terms of the physiology, then which later causing the secondary infection coming. For example, the disease, if the temperature is too hot in captivity, where the disease will come to attack the fish, and it causing the fish kill. And of course, when causing the fish kill or lower the production in terms of retarded growth of the fish in captivity, will impact the economic or the supply chain of the fish supply in the nation, in the countries. So this happen everywhere actually, where the climate change prone to happen, right? Like in our area, for example, it's very, very rampant from 10 to 10. Only thing the farmers didn't realize this, because of the farmer thought that this is normal to them. But for the fish that in the captive environment, they experienced it too much. They will suffer too much for this. So that one will affect the economy as an intangible impact of this climate change to the economic and supply chain of our foods. supply, especially in fisheries. Yes.
Okay, thank you very much. I
Yeah.
think the explanation is clear already.
Mm-mm.
Yeah. Okay, one more question, Professor Mazlan. I think
Yeah.
from the question is coming from Ismarika
Yeah,
to
okay.
Professor Mazlan. Is it any significant impact of COVID-19 on Malaysian aquaculture? And what is the government and academicians' actions to overcome this problem?
Okay, that was
Yeah.
another.
Thank you.
Sorry.
Maybe you could
Thank you for
address this
such a
question.
nice, beautiful question.
Okay.
Actually,
Go ahead.
C19, as we read from the news everywhere, for example, is Malaysia condition, I believe, quite similar to Indonesian and the Thailand, for example, yeah, where the people are urged to stay at home, right? And furthermore, through this, the fourth cycle of the COVID-19 attack due to the new variants, variant of concerns, all right, VOC, and then that caused more tight control by the MCOs, like the Majlis Kehormatan Negara, so security, national security. Then it's very, very significant impact in terms of people cannot go out from their home to go to work. They're similar to go to work in the industry lah, for example, in the pond, in the hatcheries, where people are very, very difficult to move. But on the other hand, our government allow for the fishermen to catch the fish in the sea, in the open sea, in the wild, because when they go out to the sea, they are confined to their boats. Okay, with the small crew numbers, then of course they manage to catch the fish for our food to bring it to the market, right? But for in the aquaculture industry, it's quite difficult because they are on land, and normally they are labor intensive, which of course the government also understand that the most of the aquaculture industry employs the foreigner labor workers, like from Bangladesh, from Myanmar, or things like that. That also having a big problem in terms of the disease transmission for the C19 in Malaysia due to the workplace. That's why Malaysia government strongly impose a very strict measures in order to control the entry of the workers, on sites, including in the aquaculture farm industry. So that's why this affecting very much in the aquaculture industry, because of this, unable to to harvest the the fish and prolong the harvest of the fish, or maybe the disease developed due to uncontrolled environment. Not so many people are looking for it in terms of water quality control from time to time, and in a daily basis. For example, there's heavy rain, sometimes very hot. So in terms of the oxygen supplies control, also very, very important on site to maintain the fish in captivity, also in the pond. So that is the factors why in so much from the survey that I mentioned from my slide, so many people are asking from the government for the financial assistance, for example, in order to survive, to survive for their business. All right. So it's of course no question for this. Yes, a very, very
okay. Thank you very much. I think it's clear to be addressed about the impact of COVID-19, I think, on Malaysians aquaculture.
Because people in Malaysia, they follow the rule. When government say no, minute they don't know, they going out. We're not going out.
Okay, following orders.
Yeah, following the orders. All right. Orders,
okay, once again, thank you very much, Professor
Yeah.
Mazlan, for your time. Okay, we go to the next questions. I think we could ask the Professor Yunichi. Yeah? There is one question from Irwan. Yeah. Professor Tanaka, how to maintain the balance of marine ecosystem from the exploitations of bioactive compounds from marine organisms, such as sponges and others. Okay, please go ahead.
I already wrote a short message on the chat room, but I'm going to repeat it. Nowadays, thanks to the instrumentation, we don't have to collect too much amount for chemical analysis as we used to do back in 1980s or so. So we only collect a small amount, handful amount of material just for screening, so on. Usually it is not necessary to accumulate large amount. So I hope our activities do not damage coral reef ecosystems.
Okay, I think it's a clear explanation from Professor Tanaka. Once again, thank you very much for Professor Tanaka for your time to join us here in
I enjoyed also.
Icfaes 2021. Hope we have good collaborations in the future. Okay, the next question is coming from participants to Professor Yushinta. Yeah, from Ita Apriani, permissions to ask what is the difference between phytobiotic and phytopharmaceutical? Professor Sujaya.
Yes,
Okay. You are ready to ask. You are ready to answer. The question is coming from Ita Apriani. She is asking what is the difference between a phytobiotic and phytopharmaceutical? Okay.
Okay, thank you.
Yeah.
Ibu Ita, for your questions. Actually, phytobiotic and phytopharmaceutical is the same, just different term only. Many research also called herb medicine, and in Indonesia mostly called jamu. You know,
Okay.
jamu.
Yeah, jamu. Yeah.
I think this is a new technology in aquaculture, but in the poultry, it's have been widely used. Yeah? So phytobiotic and phytopharmaceutical is the same. Have medicine, yeah, it's the same too. Thank you.
Okay, I think the question has been answered by Professor Fujaya. Okay, there is one more question, Professor Fujaya, from Julia Niwatana. Yeah, there is one more question, I think, from Julia Niwatana. He is asking, I think this trio biotic is a new term in aquaculture industry. Is there any commercial product of it? Thank you, Professor. Okay.
Yes. Okay, Ibu Julia, thank you.
Oh, Ibu Julia, sorry.
Two very good questions. Yeah. You're right. Theobiotic is a new term because we are made the term, yeah, to describe the three biotics in the one product: probiotic, prebiotic, and phytobiotic. Yeah, in the one product, and this product already launched in the limited farmers in South Sulawesi, and maybe could be tested in Aceh fish farmers, maybe, or we could join research about that substances with the researchers from UNSIA and others. Yeah. Who interesting the technology. Okay.
Okay, thank you very much, Professor Fujaya. For those, I think, who were asking the questions, perhaps there will be giveaway from the organizers. Isn't it, Chairman? Okay, ladies and gentlemen, I think we finally come to the end of this seminar. Yeah, I would like to thank to the speaker for the informative and interesting presentation, yeah, and all the participants for very active participation. Once again, please give big hand for the speakers. Okay. See you some other time. Have a nice day. Thank you.
Thank you.
Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Oh, I think there is one request from the organizer that they would like to have a photo session. I think we could open the video, so we could have a good background of Icfaes 2021 to be documented by the organizer.
Thank you, Dr. Yopi, and also keynote speakers for sharing your expertise. Ladies and gentlemen, kindly be requested for all participants to post your best position on screen as we will have a group of photo session. Yep, kindly be requested for all participants to post your best position on screen as we will have a group of photo session. Thank you.
Please open cam.
Photo session. Tadi Bu Mare WA apa kali? Intinya ada terus cari itu masalahnya lagi tahu itu jangan orang dia.
Is it done?
It's
All
done,
right, so
yeah.
thank you very much. Ladies and gentlemen, this is the end of the opening ceremony of Icfaes 2021. Everyone is invited for lunch break and Zuhur prayer until 1:30, followed by oral presentation session divided into 5 different chat rooms. For your information, link can be found through chat room and also WhatsApp group. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Professor Mazlan, Professor Yusinta, Professor Tanaka, thank you very much.
Thank you, Mr. Chairman. Most welcome.
It was very outstanding explanation.
See you. Let me leave.
In Makassar, is it rainy, Bu Yusinta?
Yeah.
Yeah, rainy.
Hi, Pak Muhammadar.
Pak Muhammadar.
How are you? Wow.
Pak Muhammadar, please speaking.
Tak terdengar.
Pak Muhammadar, nggak kedengaran.
Mic, Pak Muhammadar, mic.
Prof. Yusinta, terima kasih atas waktunya. Thank you very much.
Sama-sama, Pak. Gembira sekali diundang sama Pak Dedi.
Ya, insya Allah mudah-mudahan tahun depan bisa selesai pandemi ini, kita bisa buat
Iya.
buat ini ya, luring ya. Kurang strict rasanya.
Kurang ya.
Secara akademis sudah memenuhi syarat, tapi kita secara personal, sosial kurang kayaknya ya.
Betul Pak. Mudah-mudahan tahun depan sudah lewat nih pandemi. Sekarang di Makassar sudah semakin
kurang ya.
Ya, semakin banyak.
Oh, semakin banyak.
Oh ya.
Ya Aceh ya, alhamdulillah udah agak lumayan
agak ini ya, Makassar bahaya.
Ya, terima kasih ya sekali lagi.
Oke, sama-sama Pak. Saya pamit dulu ya.
Ya, sampai jumpa.
Sampai jumpa lagi. Terima kasih.
you.